Hi, science friends. Happy Friday. It was really good to see you all this week, most of you. We are starting our new unit in gene expression, so you should have those materials if you want to pull out your guided notes and follow along with me as we go through. So one of the last things, or people rather, that we talked about in our genetics unit was Thomas Hunt Morgan. Remember, he was the student at the University of Kentucky. He studied the fruit flies, and Thomas Hunt Morgan lived in like the mid-1800s, and he had determined that chromosomes contained the genetic information. That's where genes and traits seem to be passed down, was on the chromosomes, okay? So we know that whatever it is in the chromosomes, and that's how it gets passed from generation to generation. But beyond that, we don't really know what the information in the chromosome is. Like, what type of macromolecule is it? So when we try to answer that, and when the scientists then tried to answer that, they think, well, what are chromosomes made of? Well, if you remember, chromosomes are made up of DNA. So this is my DNA. But it coils tightly around these proteins called histones. It kind of wraps up around them like a thread on a spool. And so your DNA coils around histones to be able to form chromosomes, okay? And so when they're trying to figure out what is the genetic material inside the chromosome, they look to the DNA and they look to the proteins, because that's primarily what the chromosome is made up of. And they think, well, it must be one of these two. This is what chromosomes are made up of. So the genetic material must be DNA or proteins, okay? Well, scientists already knew that proteins had a lot of function. And so really they kind of leaned toward proteins as being the genetic material. Now that sounds crazy to us now, because that we know that DNA is the genetic material, but at the time they didn't know that. And so in the mid-1800s, the first person to do experimentation on that was a man by the name of Frederick Griffith. And he did some transformation experiments with bacteria and mice. And so what transformation means? Transformation means when one cell takes a piece of DNA from another cell and begins to express it, okay? It starts to express it both in genotype and phenotype. It assimilates, okay, or incorporates that DNA into its own DNA and then goes on to express it. And so that's what transformation is. So what Griffith did is he took some bacterial cells. So we've got two types of bacterial cells. These first two serve as the controls. So we've got an S cell, and when you infect the mouse with it, generally the mouse dies if it's a normal living S cell. Then you've got an R cell, and if you infect the mouse with the R cell, it's fine. It doesn't get sick. It stays healthy, okay? Now, this experiment may seem pretty simple to us as we go through it now, but it took him about 14 years to complete this experiment. So, this wasn't something that he did in a day. When scientists do this research, it is a lifelong endeavor, okay? So, S cells killed him, R cells, he was healthy. So what he did then next was he took heat and he used it to kill the cells. We know that when biological things are exposed to really high levels of heat, that it kills the cells, okay? And so remember, the S cells were the ones that normally killed the mice. But if he used the S cells and heat killed them, then look what happens. The mouse is healthy, okay? So whatever he did there, he did it, that genetic material, that DNA, it was inactive, okay? Now, in the next part, he took a mixture of heat-killed S cells. So remember, heat-killed S cells produce a healthy mouse. And living R cells, okay? Those also produce a healthy mouse. And so we would expect this to have a healthy mouse, right? But look what happened. The mouse died, okay? So basically what happened is these living cells right here, the R cells, took up the genetic material from the S cells. Even though those S cells weren't living, they still contained that DNA. And so that DNA was assimilated, going back to our terminology here, into the R bacterial genome and then expressed, causing the mouse to die. Okay? And so in doing this, he showed that DNA was the genetic material. And he, went through this whole process of transformation. That DNA can be uptaken by the cell and it can be later expressed and, cause death in this cell like he saw. Now, this caused an uproar in the science community because the scientists really believed proteins were the genetic material. That's just kind of what they thought, okay? So there were some other scientists that derived other experiments to be able to test this idea of is DNA or protein the genetic material. And so the next one are the Hershey-Chase experiments. That's two scientists, their last name. And so they did experiment on phage. So this is a phage here. I did a semester's worth of research in college on bacteriophage. That's all I did. Played with bacteriophage every day. So what a bacteriophage is, or abbreviated just phage, it's a virus that can only infect bacteria, okay? It has a specific host that it infects, and it's bacteria, and so it's called a bacteriophage. So this is what it looks like under like a scanning electron microscope. And so what it does here is when it infects the host, this is its tail, it injects its genetic material that usually lives up here in the nucleus into the host. And you see that there, it's DNA, into that bacterial cell, okay? So that's important for this experiment. So what Hershey and Chase did in this experiment is they used radioactive isotopes to label the different parts of the bacteriophage, okay? So this phage head and the body of the bacteriophage, those are made up of protein. But in the inside of the bacteriophage, in the nucleus, that's where the DNA is. And so they radioactively labeled them. And this is the one that contains protein, because some proteins have sulfur in them. And this is the one that contains DNA, because DNAs have P, which is phosphate or phosphorus for that phosphate group in them. And so what they were able to see is that when the virus or the phage infected the bacterial cell, you don't see the genetic material is inside. That's the part that turned blue, okay? You could detect that radioactive genetic material inside. For the phage were still on the outside of the cell, okay? Indicating that genetic information that goes on to be able to reproduce inside the cell is DNA, okay? So that's how they determined that DNA was the genetic material. So let's just review. Going back to biochemistry, DNA is a nucleic acid. That's one of the 4 types of macromolecules. Remember, the others are protein, carb, and lipid. Okay? There's 2 types of nucleic acids, DNA and RNA. So nucleic acids always have 3 parts: a 5-carbon sugar, a phosphate group, and a nitrogen base. You ought to be able to say that in your sleep. In DNA, the 5-carbon sugar is deoxyribose. In RNA, the 5-carbon sugar is ribose. The phosphate group is the same in both. And then the nitrogen bases, in DNA you have adenine, thymine, guanine, and cytosine. Okay? Now, we'll talk about one more later. In RNA, you have one other base called uracil. Okay? But we'll get into that more later. So, we touched on this briefly. And if you've had any other science biomed classes with me, then you probably know this. In DNA, the DNA nitrogen bases pair together in a certain fashion, and that's known as Chargaff's rule. So A pairs with T and G pairs with C. And so the percentage of A in a cell or in a molecule is equal to the percentage of T. Percentage of G is equal to the percentage of C. Okay, so that's within a species, the number of A and T are bases are equal, and the number of G and C bases are equal. Okay, so when we look at our DNA molecule, this is how it looks. This blue part here is our 5-carbon sugar. Sometimes it's referred to as a pentose sugar. Ose because sugars end in ose. Pent because it has 5 points like a pentagon. Okay, and then we have our phosphate group here in yellow. PO4, maybe you remember that from biochemistry, is a phosphate group. So the sugar and the phosphate alternate to make up what is referred to as the sugar-phosphate backbone. If we're looking at our DNA, remember it's a twisted ladder referred to as a double helix. This is the sugar-phosphate backbone, the sides of the ladder. Here in the middle are the nitrogen bases. Those make up the rungs of the ladder. Okay? Sugar-phosphate backbones on the outside and nitrogen bases in the middle. And then those will twist like you see here in this diagram. So this part here is going to be sugar-phosphate, sugar-phosphate, sugar-phosphate. In here it's going to be A and T and G and C and so on and so forth. Okay? Now, in RNA, the structure is the same, except you only have half of the helix. You don't have the other side of the ladder, okay? You only have T. You don't have the A over here that pairs with it, or the T or the G or the C that pairs with it. This diagram is technically more like RNA because it only shows half of the helix, okay? Now, another thing that you might notice about these A's, T's, G's, and C's, those are referred to as nitrogen bases. Okay? Thymine is a single ring structure. You see it here. Cytosine is also a single ring structure. Adenine and guanine are double ring structures, okay? So when they pair, A pairs with T and G pairs with C. So every time you have a double ring structure, pair with a single ring structure so that it's 3 rings across, okay? That's important so that our ladder stays parallel. We can't have it going, 3, 2, 3, 2, wavy in our ladder. That wouldn't work, okay? Now, I'll get to that in a second. Okay, here's what I was going to get to. In the middle of the nitrogen bases is a hydrogen bond, okay? Now remember, a hydrogen bond is an intermolecular force. It's not the strongest, right? Versus on the backbone between the sugar and the phosphate, we have a different type of bond. This could either be an ionic or a covalent bond. Which do you think it is? Maybe thinking back to chemistry would help you. If you remember, an ionic bond is a metal and a nonmetal. A covalent bond is 2 nonmetals. So these are covalent bonds, okay? So the nitrogen bases in the middle are held together by hydrogen bonds. The sugar-phosphate backbone on the outside are held together by covalent bonds. Those covalent bonds are a lot stronger. We want these hydrogen bonds to be weak because we want the DNA to be able to break apart easily so that we can replicate it, okay? But these covalent bonds on the outside have to be strong. because that part does not need to break apart. Okay? Now, there's certain distances that the bases are always apart. That's important. There's distances between each spin for the bases. That's important, but those are not numbers that you really need to know. Okay? Now, I want to point out one other thing in this diagram. Look at the orientation of the sugar and phosphate group. So here, these oxygens always are pointing up, right? Versus over here, they are pointing down. So this is referred to as the anti-parallel nature of the DNA, okay? Anti-parallel nature of the DNA. These ends of the DNA are referred to as 5' and 3'. And this is not really super important right now, but it will be very important as we go on later in the unit to understand 5' and 3'. Okay? What 5' and 3' mean are in organic chemistry, you number the carbons. So this is 3, 4, 5. Actually, I don't know what number it is. I don't know how they have them exactly laid out. But the end, yeah, this would have to be 3. 3, 4, 5. So up here, the 5 end is left at the top. Versus down here, the 3 end is left at the bottom. So that's why it's referred to as the 5' end and the 3' end. And then over here, it's opposite. The 3' end is going to be at the top, and the 5' end is going to be at the bottom. Okay? That's a lot of information, I realize. So they thought so they knew the structure of the DNA, or they knew that the DNA was made up of, or the genetic material was made up of DNA, but they still didn't know what it looked like. It wasn't till Rosalind Franklin came along, and she used X-ray crystallography to identify DNA as a double helix structure. Now, the story of Rosalind Franklin is one in science that's just really unfortunate, okay? So, Rosalind Franklin was working in a lab in London, and there were two men in a lab very nearby, Watson and Crick, that were also trying to determine the structure of the DNA molecule. And she had found it, and she had written it down in her lab journal. And the guys got in and got her images in her lab journal, and they kind of published it out from under her, okay? And she ended up dying, and they published her work and kind of claimed it as their own. And over here, well, I thought I had a picture. They ended up winning the Nobel Prize like the following year for determining the structure of the DNA as the double helix, using her work to push them in that direction. So they went on and showed it too, but they went and snooping in her lab and saw her lab journal and knew that because of her. And so she's kind of an unsung hero in science and in DNA because they stole it and they got the Nobel Prize. And she died from cancer, very likely from the X-ray exposure from her studies in DNA and trying to determine this structure. Okay? So going back here, I think I've said all of these things. Covalent bonds hold together each nucleotide. Again, those are on the backbone. The two strands are held together by hydrogen bonds between the base pairs. And then in Watson and Crick model, they showed the sugar phosphate backbones are anti-parallel, meaning they're going to point in opposite directions. Okay? Now, I mentioned just this a moment ago about the 2-ring structures and the 3-ring structures. So the 2-ring structures are cytosine, thymine, and then also that other one, uracil, that's in RNA that we spoke of a moment ago. So if you want to write uracil there, I think that's a good idea. The purine structures, those are called the pyrimidines. That's just the class of molecule that they fall under. But that term is important. You see that sometimes on tests, purines and pyrimidines, and knowing which one is which, okay? I don't know why. I always remember ag and cut. Cut is a longer word, so it goes with the pyrimidines. Ag is a shorter word, so it goes with the purines. Maybe that doesn't work for you, but it works for me, okay? The longer word has the smaller structure, so it has the small structure. The shorter word has the big structure, so it has the 3-ring structure. That's how I remember it in my head, okay? So a purine always pairs with a pyrimidine because A pairs with T and G pairs with C. And remember, as I said a while ago, that makes sure that you always stay 3 bases apart and that you don't have a wavy backbone on your DNA molecule, okay? These bonds here that are drawn in as dots, those are those hydrogen bonds that I told you about that break down when the DNA is replicated, okay, and copied. And we're going to talk about those more in depth as we go on, and those are going to be really important as well, okay? So if you all have any questions, let me know. You have a worksheet on DNA. On the bottom, there's a paragraph. I will grade that pretty rigorously, so make sure you do a good written paragraph there and don't just try to blow your way through that. So y'all have a great weekend, and I will see you next week.